great pleasure to really, put forward Professor Robin Wood as the keynote speaker, but I think it is important, although you'll be focusing entirely on tuberculosis, to recognize that Robin was actually at the New Somerset Hospital way back in 1995, 93, 95, when those antiretrovirals were first introduced, but initially the CTAC cohort. And indulge me, one more anecdote. I remember him having this box of stiffies. Now, many of you won't even know what a stiffy is, but that was all the patient material was in this, this stiffy drive. And every day, data, I'm just telling all the young researchers in the room, every day the data got entered himself onto those stiffy drives, and the data was curated by himself for his own cohort over that time. And that is the man I met way back then, and nothing has changed. As I think, except that he's gone beyond the stiffies now and enters it directly into the computer. And I would tell you that, and again, now I'm just going to share a little bit more than you want to have shared, but throughout COVID, every single morning, and the only reason I know this, for those of you who don't know that we're married to each other, otherwise I wouldn't know this. I know a lot about researchers, but yeah, anyway. Every single morning he entered exactly the new COVID cases, where they were, etc. So the man, thrives on data, what can I say? And it has been his personal journey to discover what it is about tuberculosis that has led to us having one of the biggest epidemics and the most intractable epidemics of tuberculosis anywhere in the world. And so I think you're going to hear a little bit about that journey and how far he's come. And just want to acknowledge Digby, who's joined us. Thank you for joining us. I know your days are very, very full. So Digby is the current director of the Institute of Infectious Disease Molecular Medicine, and we're really delighted to have you here, Digby. Thank you. And he is a partner, as you will hear, in this research. So.
Well, good afternoon, and thanks for the introduction. As you say, a little bit too much information, as usual. But at least I have the opportunity to talk about an obsession of mine for the last 10 years. And I think we are changing the paradigm of tuberculosis. And the title may sound a little bit strange, but I'll explain it to you. Hunting the boson was what we actually said we were doing for the first part of the last 10 years, and then finding the bummocks is something which has just occurred relatively recently, and I'll also introduce that. So I'm going to talk a little bit about the epidemiology, but particularly the lack of progress and where we are now, and looking back over 100 years of how we actually got here. And then I'll be talking about hunting the boson and finding the bummock. So the boson, if you don't know, is a fundamental particle that was predicted, its existence was predicted by quantum theory, but nobody could find it and prove its existence. So it's a theoretical concept, and that's where the parallel will come in, and yet hadn't been found for, it turns out, 40 years until it was announced that the particle had been seen in experiments performed at CERN, and 40-year search for something which was theoretically there. And then I didn't know what a bummock was until quite recently, but essentially it's the 90% of an iceberg which is lurking out of sight, but capable of great insight. And again, you'll see the parallels of this, and yet its potential for bad things to happen is exemplified by the sinking of the Titanic. So, where are we now? this is not rates, this is just number of TB cases that are identified and treated each year, and the latest data that I could find complete was 2022. So you see South Africa on the left-hand side there. That's the number of cases. This is irrespective of the population denominator. Then you look at Cape Town, and in the second column, and then you look at countries like the United States of America, United Kingdom, France, and Canada, and you can see the magnitude of the disease load that we have. And yet, when I've looked through the Medical Officer of Health records for more than 100 years, they always have a spin of optimism to them, and how we are bringing this all under control. And I think this shows that, in fact, we have failed to get it under control. Now, you may think, well, is that just because we haven't tried? And this is a slide I put together of the TB control measures that were introduced in Cape Town from 1900 onwards. And I was very much aware that people in 1900 knew that they had a big problem. You see the second line there is compulsory notification data. So the data that we can actually work with is dependent on the notification of compulsory notification. There was quite large fines for not notifying TB. But Cape Town introduced compulsory notification of TB 8 years before the United Kingdom. So it wasn't that we weren't trying. We knew there was a problem, and if you go through all those other interventions, from pasteurization to drug therapies, we actually led the way for most of those until the last one, which is we were slow to introduce antiretroviral therapy for HIV-TB, but otherwise we have been trying very hard for 100 years. So this is some data that I published a few years ago. So this really shows you the picture of how we got to where we are. So there's three populations we're looking at here. We're looking at Cape Town in red, and we're looking at London and New York in blue and green. And if you start at the top left-hand panel, you can see that we all started in much the same place. That we had, and that's looking at mortality rate. So that's the number of death certificates that had tuberculosis written on them. And you can see that in 1910, we're much the same, a little bit higher than the others. But something happened in London and New York between 1910 and 1950, before antiretroviral therapy came in, anti-tuberculosis therapy, which was introduced in the 1950s. And you can see something's different about Cape Town from the other cities, and that's before we had therapy. If you look on the left-hand side of the bottom panel, that is the mortality rate of people who are diagnosed with TB. And you can see that, again, we were pretty similar all the way through the early 1900s. But as soon as therapy came in, we actually decreased the case fatality every bit as well as the Northern Hemisphere. And I can't really see from here, but you can see that the mortality rate for untreated TB is something like 60% per year. So everybody died within 2 years, basically. And you can see how therapy came in, changed that, but it's changed it for the ones that were diagnosed with TB. So we gave therapy. It worked as well for the individuals that we gave therapy to as anywhere else. But now you go to the top panel on the right-hand side, and you see the incidence rate. And again, the incidence rate in New York and London dropped rapidly in the early part of the 1900s before therapy came in. But in the red line, you see, it didn't change very much. It dipped a little bit in the 1950s and 60s, and then it exploded around about the late 1990s with the HIV epidemic. So I see that pouring gasoline on the fire was sort of a talk, I think, this morning from Karen. So a situation where we're failing totally to control TB. When HIV came in, it just doubled everything. And the benefits that we're seeing at the moment in decreases in incidence rate are probably related to reversal of that with antiretrovirals. So then, again, we'll go down to the bottom right-hand panel. And what I'm looking there is I'm looking at the TB rates in children, so between the ages of 0 and 4 years. And again, you can see that in the red line, there's no change, whereas there's a steady change all the way through from 1900 until the present day in New York and London. Not much difference because of antiretroviral therapy. It looks like it's a straight line over the period of time. And the reason we're looking at those is that children are a measure of ongoing new infections, because they haven't lived long enough to have the disease and then reactivate subsequently. And then, not to take this point too much, but here I'm just comparing Cape Town and New York. It's very similar data for London. It's just that New York data is more easily available. I had to go through a lot of libraries in London to pull out the data. But what you can see, and this is slightly different from the other graphs, this is a log graph on the left-hand side on the y-axis. And you can see essentially, if you look at the whole lifetime of individuals and you look at the rate at each age group, you can see again that in Cape Town, there's been no difference. There's a decrease in TB in the early teens, and that's known as the golden era of TB. But otherwise, there's been no improvement. If you look at some of the faint lines at the top of the 20 to 30-year-olds in Cape Town, that has increased, and that's the reflection of the HIV epidemic. But you look at New York, it started off with exactly the same distribution, but it's become a disease of older adults. Children don't get it. That tells you that the transmission has been interfered with and stopped in New York, but I don't believe we've ever done anything to reduce the transmission to any significant degree in Cape Town. So what can we learn from that? In the 1910 to 1950, there's obviously a difference which is not related to therapy between the two, the north and south. TB mortality remained very high in Cape Town, but it dropped like a stone in the Northern Hemisphere. In the 1950s, TB treatment decreased case fatality, and we did just as well in Cape Town as the Northern Hemisphere. But TB transmission remained high in Cape Town, although it almost disappeared in the Northern Hemisphere. And then we can look, and the coincidence of the explosion of new cases with the HIV epidemic seems as though that's immunosuppression coming into a population that TB is not under control. So, why could it be that transmission remains high in our settings? And essentially, I think there's three sort of hypotheses. The one which is very popular in the Northern Hemisphere is all you have to do is identify the TB positive, smear positive cases and treat them properly. I've had arguments with people who put forward this argument for the WHO, etc., and they just say, well, you're not treating people properly. And I think that's untrue. I think we are treating people well. Case fatality drops, but we're not getting rid of the transmission. And if you look at smear-positive cases and try and look at transmission to other cases, you can only look at people who develop smear-positive disease and compare those two bacteria to show they're the same. We can only find about a third of our transmission is from people who are identified as sputum-positive TB. The second hypothesis, which is sort of becoming a little bit more popular now, is, well, maybe there's a whole load of people in the community that have sputum-positive TB, but they don't present. So let's go and look for them. And I think that's probably the emphasis on TB control program that's current. I didn't show you here, but in fact, between 1948 and 1994, Cape Town had a mass miniature radiography service, which was very good. It was stopped, again, on the recommendations of the WHO that say you should spend your time on treating people, not finding these. But it worked very well, and I think that might have been responsible for that slight decline in the 50s and 60s. But what I'd like to put forward is the third option, which is what has really motivated my research for the last 10 years. And this is what I'm calling hunting the boson. That could there be a previously unrecognized reservoir of transmitters in our communities? Certainly, if this was the quantum theory of TB, you'd say, well, there's a good possibility of that, but no one's found it. What are we looking for? We're looking for a TB bug in the exhaled breath, because we know it's an airborne disease that transfers from somebody with the infection to somebody else that's the new susceptible. And we're looking for something which is a few millionths of a meter long. And if you look on the right-hand side, you see how many of those type of units fit onto the tip of a needle. And so we are looking for less than a needle in a haystack, hence the hunting the boson. So a few years ago, we had this idea that let's try and see what's coming out of people. And this is an interesting machine. This is a machine that I got from Finland. And it's a machine that's designed to measure the number of particles coming out of a diesel engine. So what it does is it impacts those particles onto an aluminium plate. And this was one of those plates. And it was scanned by Jeremy Woodward from the physics department here with his electron microscope. And he spent a long time looking for this needle in a haystack. And he came up with this. And we found what we believe is the first picture of a TB bacillus that has been exhaled in the air. Digby's in the audience, and he said to me that this looked like the poster boy of TB bacteria. So the question is, was this the first hint that we might be finding the boson? So we wanted to search for these organisms, and the place to start is, well, let's look at people with smear-positive TB. And we developed a system for capturing, at that time, coughs as well as other respiratory activities, because there's been a prejudice that cough, which is associated with severe disease, might be responsible for transmission. So that meant that we had to capture coughs, and coughs come out very, very fast. They come out at 40 meters per second. It's very tricky to... So we worked with some computational fluid dynamic modelers in the University of Cape Town, and worked out what speed we needed to draw the air through in order to capture these. And this is essentially what we did. And then once we've got that air going through, we can monitor the amount of carbon dioxide in there, which is telling you how much air the person is exhaling, because we are metabolizing food and producing CO2 and water. We can look at the particles, and we can try and collect those particles and look at them for TB imaging a little bit more efficiently than the diesel motor monitor, and look at non-volatile organic compounds that might be present in those droplets. So this is actually 10 deep breaths of my own. You can see in the blue at the bottom that each time I breathed out, the carbon dioxide level goes up and down quite nicely, and that allows you to look at what's happening to the particles. And the particles are very small ones at the top. The squiggly line in blue is large particles, which are more than 5 microns in size. We think that TB is in the middle portion there because it's about 1 to 2 microns in length. So we're looking at those. But it shows, in principle, that we're capturing the CO2, we're capturing the particles. And this is in the unit down at Massey, we made this respiratory aerosol sampling chamber, and you can see somebody wearing a Tyvek suit, breathing into this funnel with air being pulled through very rapidly. And you can see the supervision of our nursing staff. And this is what we're measuring. So on the right-hand side is a particle measurer producing the data that I just showed you from my own respiratory activities. Then you can see that we're collecting into a wet-walled cyclone. We have a pump which is variable speed, so we can adjust the speed, and we can measure in the little rectangle thing on the left-hand side, measures the temperature, humidity, and the environment that people are in. And then this is just a little bit closer. May look easy, but every one of these parts has to be manufactured locally in order that it can be sterilized totally between each patient, so there's no risk of any transfer of active TB from one person to another. And it's all made out of medical grade stainless steel. So it's one of the things I like doing, is playing with expensive machines, so this is one of the things that I do. So that's the collection, but we need to detect the organism. How can we improve TB detection? And this is something that sounds very complicated, solvatochromic trailose probe. It was developed and published in the Scientific Translational Medicine in 2018. Caroline Bertozzi was probably the person who developed this initially, and she won the Nobel Prize for chemistry a few years ago. But essentially what this is it's a foodstuff. It's sugar, which you feed to the TB organism, and on it is a little star fluorescent marker, and that gets processed, eaten by the organism, and then incorporated into the growing cell wall. So it tells you that it's an organism, it's metabolizing the sugar, and the cell wall is growing. The slide has got a little bit messed up, but left-hand side is just showing you there's a few other organisms of the same family of organisms that might be positive for this besides MTB itself. And on the right-hand side is trying to explain what a solvatochromic probe is, and what it really means is that in an aqueous setting, it doesn't fluoresce. But if you have no water and you're in the lipid envelope of the TB bug, then it fluoresces a lot. And this is how we detect the organisms. We have a fluorescent microscope. So it means that our threshold for diagnosis of an organism being present is 1. So if you find it and it looks like TB, then that's the level, which is totally different from every other way of measuring number of organisms, which normally is done by colonies of tens of thousands of organisms or hundreds of organisms messed up and looking at their DNA. This is actually looking directly at the organism. We also developed this little slide nanowell on the right-hand side, which shows that the statistics of an organism getting in there and being fluorescent, you're not likely to get multiple ones in one of these wells because there's thousands of these wells on the slide. But what it means is if there was any background noise, that it gets distributed to all the others. So your signal-to-noise ratio is much, much greater. And this is some work that was done in the IDM, and it shows very nicely the organisms taking up this dye, which is a green fluorescent dye, and you can see it's getting concentrated generally in the poles of the organism, which tells you that that's where it's growing. On the right-hand side, you can look at the intensity of this fluorescence all the way along, and that length is 1 bacteria. You can see that generally it's fluorescent at each pole, there are a few that are fluorescent in the middle, and those are organisms that are about to divide. And it shows you that you can actually describe each individual organism by its length, its width, and its shape and size. So now we've got a very efficient collection system, sampling system, and we have an incredibly sensitive detection system. So now it's time to move into some clinical studies to find out what's happening in the exhaled air of individuals. So we started with clinic attendees. Left-hand side looks a little bit complicated, but essentially people go into a TB clinic because they think they might have TB. The clinic processes them and says to some of them, yes, you've got smear-positive TB. It says to some other people, we think you've got TB, but the smear isn't positive, but your chest X-ray or your urine lamb looks positive, so we're going to treat you. So Group A and Group B on the left-hand side are people who are diagnosed with TB and get treated. And there are other people who don't get diagnosed. And they may have a broad-spectrum antibiotic and then sent home without a TB diagnosis. So what we did is we took all three of those groups and we followed them equally. When we started this study, we didn't expect the results that we got. We just expected to find them all in the smear positive, and we hoped we'd find some in the smear negative TB. Just on the right-hand side is the characteristics of the populations we looked at, and essentially there's not much difference between them, but whoops-a-daisy. Yeah, I've highlighted in red that the smear-negative TB cases had a very high prevalence of HIV infection. So this is the data that we got. So if you look at the number of organisms on the left-hand side that we could calculate in these patients, and you can see that there's not much difference between the A's, B's, but we were gobsmacked that we were finding them in the patients that came to the clinic and were discharged without a TB diagnosis. On the right-hand side in panel B is just showing you the proportion of positivity we got, and you can see it's over 90%. As I said, we followed these patients during the 6 months of their treatment. So the purple and blue are receiving TB treatment, and the yellow are the people who normally would be discharged, but we're following them up in exactly the same way. And you can see, again, we were gobsmacked. It started very high, and it went down very similarly in all three groups. So we have a very high carriage at baseline. At the end of treatment, the fourth visit, which is 6 months, still we can find the organisms in about 20% of people, but there were very small numbers of organisms. So not only did the prevalence go down, but the number of organisms went. But what it shows you is that we have some way of measuring at the end of treatment how successful it was at clearing this, this pool of potential infection. So this is showing the proportion of bioaerosol positive over the 6 months on the top left. And we scratched our heads to say, well, what's going on here? It can't just be treatment, because it's happening in the group that aren't being treated as well as the group that are being treated. So under those circumstances, we looked at what does this pattern look like? And on the bottom left, you can see the resolution of symptoms. And sure enough, when we did a linear effects model, we showed that the clearance was best predicted by the number of symptoms that they had. And what we're hypothesizing is that the symptoms are caused by the immune recognition of these cells. So we think this is an immune response, and that's what causes the symptoms. And when you get an immune response, you can clear the organisms, both in the treated and untreated groups. So next stage is, well, these people are coming out of the community and going back into the community. Let's have a look at the community and see what's happening with their carriage of the organisms. I should have mentioned the last data was published just recently in the Proceedings of the National Academy of Sciences, and this data was published in iScience. So this is a paper first author is Ryan Dinkeley, and what he's showing is that there's a pictogram of Massapequa on the top left. We got all the earth numbers and then had a random selection system for randomly selecting people who lived on those earths. Then they would come in, we would do the RASC sampling, as you can see there with the funnel, and then we can look at how many of them had TB, classically with symptoms and a sputum positive, and how many were bioaerosol positive. And in this study, Ryan looked at them at baseline, 2 weeks and 2 months, exactly the same time period as we looked at the TB attendees, so that we will eventually be able to compare those. And this is what he found. So if you look at the top, these are the number of organisms on 3 visits, and you can see in purple, most people have very low numbers, but they remain about the same. So unlike our clinic population, which went down, they stayed the same. 1 or 2 of them started low, went high, started low, went high and down, and et cetera, et cetera. But the majority of them stayed much the same. And if you look at the bottom left-hand panel, you'll see the prevalence of a carriage at each visit, and it's much the same. And there's a very interesting graph that Ryan produces to show essentially that these organisms remain much the same at each visit. And you can see just above there the medians of these didn't change. So this is some unpublished data, but I think it illustrates the point. So the first 3 panels, sputum positive, sputum negative TB, suspected TB, the purple is the first visit, and you can see the graphs move over to the left, and that means that the number of organisms are going down. And that's what I showed you over the 6 months of follow-up. The healthy controls, that's not the case. These are not symptomatic patients. They remain carrying small numbers of organisms, and it doesn't change significantly over time. So we think we might have discovered the bummock, and we think that we have evidence that there is a hidden population of potential transmitters that may constitute this. And the bummock quote came from Sarah Fortune, who's a professor at Harvard University and was writing a commentary about our paper. And she brought in this rather nice analogy with the Titanic question. And I'll just read the last line, what she said. Individuals infected with MTB, but who do not meet the current clinical criteria for active TB, but there is emerging evidence of TB carriage in their exhaled alarm, and that this is raising alarm bells for TB control. So if you put this together, and this is a little model that Digby and I put together, if you look at the 3 boxes on the left-hand side, it represents the Massapequa community. And we have a minority of people who are bioaerosol negative, about 20-25%. We have about 60 to 80% who are bioaerosol positive, and then we have this 5% that are presenting to the TB clinic with symptoms. And what we note is the people who go to the clinic, spontaneous, because they're symptomatic, spontaneously clear. 80% of them go to negative, 20% partially clear. And this is a different model from the current paradigm, in that we're taking transmission away from the definition that they have to be sick patients. We know with COVID, not only the known COVID patients transmit, but other people transmit, and that's pretty true for most diseases. There's asymptomatic carriage, and that's why we can't control epidemics very well. So what we're saying is the paradigm for TB was you get one organism, you're going to go down with disease. We don't think that's true. Interestingly, we looked at quantiferon, which is the measure of immune recognition of TB, and it didn't vary between the ones that bioaerosol, negative or positive, but it did go up with the ones that produced an immune response and either developed symptoms or disease. So what we're saying is the bummock is about 90% of the population, whereas previously we only thought the smear-positive TB cases were transmitting. And I think this may well explain our failure to control TB here. So the next thing is, okay, you've identified this group of people. It's possible that we could target them with either drugs or with vaccines. If they can clear the organism on their own, then reproducing that immune system response would be very good. But how the heck are we going to find these people? Because we can't have the sophistication of our RASC everywhere. So we developed a new system, a new sample called the exhaled breath condensate collector. And essentially what this does, it takes breath that you breathed out, and the breath coming from low down in the lungs is 100% humidity at around about 37 degrees temperature. If you cool that down, then you precipitate the liquid. And the air carrying capacity of air at low temperature is very low. So you can collect samples. This is a machine that we've made. It's a little desktop thing the size of an A4, but about 10 centimeters high. Have a disposable collection system straight into a tapered glass detector. And we've looked at the same staining, and you can see here that we can find the same type of organisms in the exhaled breath condensate that we were finding in the respiratory chamber. And then interestingly, we used the LAM, which is one of the tests that's used in HIV-TB in urine to diagnose people with very advanced HIV. We're finding this structural component of the organism very commonly in TB patients and in other people, and we need to work on that to try and understand that a little bit better. But you just take some of this liquid, put it onto a lateral flow assay, and get a response, a result within about 8 minutes. So this is just to show you where this work takes place. This is the Aerosol Research Center at Massachusetts, and our labs and the clinical spaces are there. And I'd like to just finish up with acknowledging that this is the work of many people. So at the top, we have the people within our ARC Center in the foundation. Second, we have Ryan and Digby and his team from the IDM, the help of other people from electronics and from physics, and Gilla from UCT. And then another interesting group from Zeteo Technology. They are people who've worked for the US looking at bioterrorism and developing some of the devices to identify that. They've been very helpful. Then we have the team at the University of Amsterdam, Stanford, as I mentioned, some collaborators there. And we are part of something called the TBRU, which is led by Branch Moody at Harvard and Q Ree at Cornell. And you can see, and also Adrian Minar, who's making our DMN tray staining at the University of Groningen. So those are the collaborators. The funders on the bottom left are the NIH, Gates Foundation, and the European Union, and then the local support from the NRF, Department of Trade and Industry, and the South African Medical Research Council. So thank you for your attendance. And we've got a few minutes left if people want to ask questions.
Yeah, I wondered this one we felt really did warrant questions, so if people do have any particular questions or comments or thoughts that they would like to raise, we have a few minutes. Yes.
Thank you so much for this really, really interesting presentation.
I think it's absolutely valid comment or criticism that this needs to be reproduced in other populations. I wouldn't choose Constantia. The reason for that is we are living in a sea of TB, and your assumption is that the wealthy are not getting this, and we have the same TB rate as New York and but there are other factors at play here. But it does need to be reproduced. We're talking to people doing it in KZN. There's another interesting idea to do it in Uganda and London. So certainly it needs to be reproduced. What it's really saying is that some people can be colonized. And I don't know that if you're rich, you might not be colonized. There's something different that makes you have disease. So what I'm postulating is that the disease is the immune response to recognizing this organism, but that you can clear it. We have done visitors that visit us from the States, and certainly they have lower or zero numbers of organisms, so you may be right. But it does need to be reproduced elsewhere, but it needs to be linked to real transmission. When we model, and Benjamin has got a modeling paper out, the number of times that you're exposed as opposed to the number of times you're infected, it seems like it's scores per year. So there's a turnover which is very high. But obviously what changes people from being colonized to being recognized by the immune system and getting disease is key. I'm fairly optimistic that that clearance in Group C, if we can reproduce that at a population level, it's a completely new vaccine target. And it's a vaccine targeted at transmission rather than disease. So I think that's the optimism of this. And that's really why we thought, well, how the hell can we find these people? And that's why we need a very cheap, easy way of finding them. But you're right, this is one site. Everybody says there's something special about Cape Town, but it needs to be reproduced elsewhere. And we couldn't do that whilst we had that incredibly high technology demanding thing. We tried to do it elsewhere in Worcester, etc. We can sort of get the idea that it's working in Worcester, but they can't sterilize the things like we do, etc. So it's getting it more low tech is really important.
Thank you. Other hand. Was it Steve? Yep, Steve.
This is paradigm shifting work, but just logic. Tell me if this is if I'm understanding you correctly. So here's what all but 5 of us have got carrying a live active TB. Very few of us are likely to get sick. And so we probably represent your group C, those people who...
No. The C is asymptomatic. It's the controls that you'll represent.
Right, okay, so there will be controls. But your hypothesis is that TB can still be passed between us and still not make any of us sick until it gets to someone who has some sort of vulnerability, some sort of predisposition to getting sick. Either their immune system's overactive or underactive.
Or a co-infection with something else. Yeah.
Something like that. Which makes me wonder, surely then the answer is about a vaccine to kill off the TB, about the social determinants of disease. The reason I'm not going to get sick with TB is because I'm relatively well off, and I'm healthy, and I eat healthy food,
And that's what happened in New York and London from 1910 to 1950. So that brought down those rates dramatically without treatment. So, and without a vaccine, yeah. So what I'm interested in is really sort of on the other question is, did the West get rid of the organism? Or did they just change the host? And it'd be interesting to reproduce this work, and Benjamin's coming next week and wants to do it in the East End of London, and it'd be fascinating. I don't know what the answer is going to be, because it could be that everything's due to the factors you're saying. That's, meat consumption. If you draw lines of the TB control against meat consumption for those countries, they're very highly linked. So yeah, I mean, all I've done is found the bummock, but the disease is still driven by factors which you're intimating. Yeah.
maybe climate change will take care of the blummocks. Millicent.
Thank you very much, Prof. This is really groundbreaking. I'm just sort of wondering, get information around vaccination, saying that child, when you're a child, and as the case, have received vaccination, that they actually received.
So our vaccination policy is aimed at decreasing serious disease in children, and I think it does that. It does not change TB rates in 20-year-olds. We found no evidence for that. So, I mean, it's that devastating TB for very young children that BCG at birth helps. When I go back, when I was at school in the UK, at the age of 13, we all had a skin test, and if we were positive, you get sent off for x-rays, and if you were negative, you got BCG at the age of 13. And if you look at the sort of world data on that, it looks as though it seemed to work the further away from the equator you were. There's a lot of debate about how well the studies were run in India, et cetera, what endpoints they were using. But generally, I think it worked in the UK, but TB became so rare that it wasn't worth the investment of doing all that stuff. So I think BCG at birth is good for children. At an older age, it probably did work reasonably well, but it seemed to be geographically dependent.